Hello, everyone. My name is Brian, and I'm a senior undergrad in Dr. Dahesh's lab in the Department of Botany and Plant Sciences in the University of California, Riverside. And I'll be focusing on the identification of calcium channels involved with MEcPP triggered alteration of calcium signature. So when you think of bacteria and plants, you don't really think they have much in common, as plants are eukaryotic cells, which are multicellular, while bacteria are prokaryotes, which are unicellular. However, they have much more in common than you think. Specifically, plant cells contain an organelle called chloroplast that allows plants to perform photosynthesis, which are descendants of an ancient photosynthetic bacteria which was engulfed. As a result, bacteria and plant cells share many proteins and pathways to perform their cellular function. Specifically, our lab is interested in MEP pathway, which is shared between bacteria and plant cells. The MEP pathway allows cells to synthesize precursor molecules to make compounds which are essential to the cell. The intermediate molecule in the MEP pathway of interest for our lab is called the MEcPP, highlighted in the yellow. Previously, our lab shown that when MEcPP accumulates, it's able to induce stress response in plants through a protein named CAMDA3 in a calcium-dependent manner. Recently, our lab has found that accumulation of MEcPP resulted in rapid influx of calcium into cells in plants. This raises the question, what is responsible for regulating this calcium influx due to MEcPP? And our goal is to identify the calcium channel and other interacting proteins involved in this mechanism. Since the MEP pathway is conserved between bacteria and plants, we first asked if the accumulation of MEcPP leading to calcium influx also occurs in bacteria, and if it does, participate in the signaling mechanism. To accomplish this, we're in the process of generating a bacterial immune that has a gene knocked out, causing the accumulation of MEcPP alongside a calcium sensor. The calcium sensor, shown in the bottom of this slide, works when the calcium binds to it, it glows or fluoresces, allowing us to detect the level of calcium in this bacteria, and as a result of the accumulation of MEcPP. For our project, when calcium is present, it causes the bacteria to appear pinkish, as seen in the test tubes. For future plants, we're trying to use what is called a genetic screen, which uses a transposon mutagenesis, which is, in simpler terms, a jumping gene that can insert itself into the genome. This will allow us to generate a pool of different mutants, which each one containing a different mutation in a different gene, which will allow us to identify which gene is involved in the calcium influx. The benefit of using bacteria to study this conserved pathway between plants and bacteria is that bacteria genome is much smaller than plants, allowing us to save us time and material when screening through these genes. Potential future application of this project include new stress-tolerant crops and new antibiotics to fight harmful bacteria. Environmental conditions such as drought, heat, cold, and nutrition deficiencies will continue to limit the growth and yield of plants, which is the bedrock of the food supply, from vegetables in our salad to the meat on our dining room table. In addition, as the population of this planet continues to grow, the demand for food will increase, putting further strain on the global food supply. Identifying the calcium channel involved in the stress response due to accumulation of MEcPP will allow us to decipher the mechanism regulating stress response in plants and develop more stress-tolerant crops to help improve global food security. Additionally, there has been a rise of antibiotic resistance bacteria when the bacteria have become more adapted to the use of our current antibiotics. This poses a grave danger to the global population, as it can lead to a rise of currently treatable diseases. The potential calcium channel we're trying to identify can be a potential target for new antibiotics, allowing us to expand our current toolbox to combat this growing threat. And I would like to thank everyone in the Dahesh Lab and along with all the sponsors for this competition.